Thank you also from my side. I have the pleasure to introduce the next two speakers in the second. We are still on the impact of biotechnology and molecular biology. The next field will be the medical side, and the lecture is given by Dr. Stylianos Antonarakis from University of Geneva. It's my pleasure to introduce him. The high technology is still in place. Dr. Antonarakis is Professor and Chairman of Genetic Medicine in Geneva, the Director of the IG3 Institute of Genetics and Genomics. Most of us probably know him very well by his numerous publications and academic rewards. He had his education especially after studying in Athens at Johns Hopkins School of Medicine in human genetics, and then spent more than 30 years now in Geneva understanding the molecular basis of monogenic disorders and complex genetics, being both a physician and molecular geneticist. Of course, he is one of the most cited persons in that field, and I don't want to make this story longer by counting all your awards. We are looking forward to what you have to say. Thank you very much and welcome.
It is a great pleasure for me to be here, and of course my lecture will not be as sophisticated and as important as the previous one. I'll try to be more practical, talking about medicine, and my understanding was that the distinguished audience is not highly populated with biologists, so I apologize if my presentation is too basic. So I would like to continue in the same vein on genomes, evolution, and medicine, and try to convince you that we cannot understand medicine without the light of evolution. And before I continue, I'd like to thank Professor Nusch, if I make it correctly, and all the other organizers for the kind invitation. And I also like to thank the funding agencies that support our research, the Swiss National Science Foundation, the European Union, a certain organization in the European Union that's called ERC, the American NIH, and 4 foundations, Ludwig, Gebert, Lejeune, and Child Care, for supporting our research. I'd like to start with a futuristic view. Antonio in Tempest, in Shakespeare's plays, said what is in a scene, what is past is a prologue. And because of this statement, I will not dwell so much in the history, and I will not mention names of scientists that participated in pioneer research. I will take the past as a collective effort of prominent and non-prominent scientists, and I tell you where we are now with the medical thinking and what we think for the future. Now, let me give you the problem. Problem is that people expect a lot from the knowledge of the genome. We are in an election year in the United States, but I bring your attention to the previous elections of Senator Obama at the time and Senator McCain at the time. And in November 2008, the election month, there was people saw the publication in the most prominent medical journal, the New England Journal of Medicine, by a biologist, Robert Greene from Harvard, and a bioethicist, lawyer, George Annas, who wrote that it would be nice from now on to release information on the parts of their genomes of the candidates, so we, the public, would be better informed of these personalities and their potential, and our votes will be more informed because we know their genomes. And in the rest of my talk, I'll try to dwell on this discussion and see if that is achievable today or that's a futuristic aspect. Now, why the question was asked? The question was asked because the genetic medicine today is at a crossroad. We are at the final lap of what we can do with the genome, because we can read the genome now quite cheaply. And we are also in the starting line because the reading of the genome offers us new opportunities and new directions in which God knows or chance knows where medicine will go. And the human genome is known. The common genomic variation is known. The rare genomic variation in each one of us will be known in the next 5 years. The genome function will be with us for the next 50 years, let's say. And what is the question? The question is the matchmaking. It's a simple game between the genome elements and the phenotypic space. Genome, let's say that's a space here with the squares, which are all the functional genomic elements in the genome, 20,000 protein coding genes and many other functional elements. And that space, the phenotypic space, all the disorders and the traits that we have. Of course, we don't have a number for that. And the future will be to play that game, to associate phenotypes with functional genomic elements through the mutations and the variants. The game is not very simple. The rare cases where one phenotype is related to one functional genomic element, but most of the time, one phenotype is due to many functional genomic elements and vice versa. One element in the genome influences more than one phenotypes. So in order to play that game, the scientific community in the 80s had a major debate. So we sequence the human genome or not. The project to sequence the genome started 1st of October 1990 because $200 million were put on the table from the American NIH. And there was an international project that took 14 years, essentially completed in 2004. The project was always driven by medicine, and the goals were to map, to clone, and to sequence the human genome. And in between the lines, goal also was to identify the variation in humans. The product of that project, in several people's views, is similar to the product of the anatomists of the 16th century. As you know, the correct human anatomy was first published by Andreas Vesalius and his colleagues in working at the time in Padova, Italy. And they published that fantastic book in Basel, 1543, De Humani Corporis Fabrica, in which they had laid down the correct anatomy of the human body. And that book was tremendously important in medicine because, as you realize, 450 years later, people continue to make discoveries in physiology, surgery, based on the correct anatomy of the genome. So think of the anatomy of the human genome, all the DNA that is in our chromosomes. Think of it as a similar achievement, that fantastic book of knowledge with 4 letters, A, C, G, and T. You see about 10,000 letters now of my genome. The genome is 300 times bigger than this, a fantastic text in which the functional analysis of it is very preliminary and we know very little. What is functional in the genome? That's the first question. That's difficult to answer. The first line of answer came from the comparison of our genome to others. Comparison is a well-known method in science. As you know, Jean-François Champollion in 1818, when he deciphered the hieroglyphics, he did this because he compared the ancient Greek text with the hieroglyphic text, and by analogy, he deciphered the hieroglyphics, and he wrote to his brother that everything that I did was the result of comparison. If you compare the human genome up in top with that of genomes of other mammals, because as you've heard, we think we have a common ancestor about 150 million years ago. Everything that's conserved since that time is likely to be functional, and everything that's not conserved we don't know. So what is conserved are the protein coding genes shown here in red, and other elements that we call CNCs, conserved non-coding elements, and those are really conserved, almost identical. So that first real estate of the genome that's conserved is probably very functional. How much is this? About 5% of our genome. Like 1.5% are the protein coding genes, and 1 to 3%, depending on the criteria for conservation, is also probably functional. Only the conserved fraction of the genome is functional. Certainly not. With a recent ENCODE project that was concluded about a month ago, and there was 6 papers in Nature that you've probably seen, from the entire genome that is 3 billion nucleotides, about 1 billion nucleotides has a biochemical reaction attached to it that makes it likely functional, either a chip sequence or transcription activity or other biochemical tests. And you see that's the fraction of the conserved part of the genome. So the majority of the conserved is biochemically active by ENCODE, but there are a large fraction of the genome, about 1 billion base pairs, that is likely functional, but with perhaps not all of it is. So the bottom line is that today about 1/3 of the genome has some information for functionality, and that's 1/3 of the genome is what interests the medical field. So let's call it the medical genome. All right, so for a general audience, if each letter is if each nucleotide of the genome is 1 letter of the Encyclopedia Britannica, as Professor Harbour said, you need about 140, 150 books to write this down. And remarkably, we have 2 copies of our genome, one from our dad and one from our mom. And I'm talking to you for about 10 minutes now, and millions of my cells have been duplicated in my blood and gut and other organs. So during this 10 minutes, I copied unconsciously all this genome and pass it from one cell to another. And the copying mechanism is very faithful, but not absolutely correct. And because it's not absolutely correct, we introduce, every time we replicate our DNA, we introduce variability. What's this variability? Variability could be qualitative, as you've heard, nucleotide substitutions, quantitative, the copy number variation, and some position alterations, which is a piece of DNA that are in one position on me and another position in you. So the most common variation in the genome is the single nucleotide changes. So here is a piece of DNA, the genome taken at random that has a C in this position, and another genome taken at random has a T in that position. We call it single nucleotide polymorphisms. How many of those we have? If I take the same piece of my genome that I showed you before, 1 in every 1,000 letters, 1 in 1,000 nucleotides vary between 2 randomly chosen genomes. So the genome that I received from my mom and the genome that I received from my dad differ in about 3 million letters. And so because of this, we are all different from each other. Not only that, this is the part of the genome that I received from my dad, and he gave me 1 copy of this piece, and the same part of the genome that I received from my mom, she gave me 2 copies of this, and so that we call copy number variation. How much of that we have? About 1,000 sites in our genome between 2 randomly chosen genomes varies in length. We call this copy number variation. If we put this together, we all are the same because we all belong to the same species. 99.1% we're identical. However, we're all different from each other, of course. And the difference is 0.9%. And we aficionados, we divide this into common and rare variation. Nevertheless, the difference that we all have makes us an interesting society. We're all different from each other, and that's a gift of nature that we're all different from each other, because, as you've heard, by being different, we have the potential for evolution. Of course, because we have this gift, as you know, in life, nothing is free. There's no free lunch. We pay a price for this. And the price that we pay for this is that sometimes this variation is pathogenic. And we have all these human disorders that we call them genetic or genomic, the Mendelian, the simple ones, due to a variant in one gene, the chromosomal, complex ones in which big pieces of our genome are rearranged or deleted or duplicated, the multifactorial, the ones that are due to many different genes and the interaction of the environment, and all somatic variants that give rise to cancer. All cancer is a genomic disorder of somatic cells. A myriad of disorders and human suffering is due to variation in the genomes. Everything that I told you is available and free in the databases. So if you go home tonight and you want to amuse yourself in a different environment than Facebook or Twitter, or looking at the movies, you can take a look at these two sites and look at the human genome and all the variation and all the functional analysis and a lot of interesting and amusing information that you can spend your evenings with and your weekends in the Alps. So the DNA sequence variants, though, they come in three flavors in medical terms. I apologize if in evolutionary terms I don't touch that much. A variation that we call pathogenic, rare, that gives us severe disorders. We know most of it. In the past 20 years, most of the work was to identify the pathogenic variants in humans, the severe pathogenic variants. The second class, we call it predisposing, common variants that you and I have a lot of them, but not absolutely innocent. The fitness of each one of these is not one, as we say. Some of them predispose us to disorders, and some of them protects us from disorder, a little bit, each one of them. And the third class is what we call neutral, really neutral, a lot of variation in our genome that plays no role in the development of our lives or the susceptibility to disorders. And the major challenge for the geneticist in the next 30, 40, 50 years is to distinguish which variation is neutral and which variation is predisposing. A difficult task, because the penetrance of these predisposing variants is very low and difficult to identify. And another important thing in genetic medicine is that each one of us has his own genome, and each one of us has his own reaction or interaction with the pathogenic variants. As you know, our species went out from Africa and populated first the southern part of the world, and then about 40,000 years we came to Europe, and very recently, like 15,000 years, populated that part of the world. But in that movement, people took with them one thing. They took their genomes, and they isolated themselves one from another, and accumulated mutations. So the genomes today are not, as a group, they are slightly different from one part of the world to another. So that's why some disorders are more common in some populations and not in others. So we need also to see genetic medicine with the eye of the geography and the eye of the history of each one of us. So up to now, we talked about the possibility for errors, for possibility to evolve, and the obvious secret in medicine is that the genomic disorders is the price to pay for the potential of evolution and adaptation. And as you understand very well, it is impossible to understand medicine and why, the why question that the patients ask us all the time, it is impossible to understand without the evolutionary thinking in the light of evolution. So the human genome sequence that has achieved, it gives us many benefits, future and present. We understand the majority of monogenic disorders. We hope we understand the polygenic phenotypes. We hope we understand the various cancers. We definitely provide accurate diagnosis, would determine the predisposition to disorders, and we have novel opportunities for treatments and opportunities for what is called individualized medicine. Most of the progress that has been made so far is with the monogenic disorders. The teachers are the patients, of course. How many monogenic disorders we know? If you take the space of the protein coding genes, which as of 3rd of October was 20,070, out of these 20,070, in the databases there are 2,859 genes that have variants, pathogenic variants that affect our health in a major way and they produce monogenic disorders. So all the sophistication and mechanisms of medicine that we know today is based on these 2,800 genes, and it is up to the next generation of physician scientists to fill up this camembert and understand more who we are. Let me give you an idea about the cost. When the genome was sequenced first time, the cost was about $1 per nucleotide. So the total cost was $3 billion. Spread over 14 years, of course, throughout all the industrialized world, the United States, the European Union as a union, France, England, Germany, a little bit of Switzerland, China, Japan, Canada. So all the industrialized world participated. And today, if you ask me, I like to have my genome sequenced, how much you pay? You pay probably about $5,000 and you wait for about 6 weeks. And if you want to sequence your protein coding genes only, the 20,000, you put $1,000 in the table, wait for about a week and a half, and you have the list of variants in your genome. And the goal is, in the next 2 years, I hope no more than this, the cost of complete sequence of the genome, about $1,000 in a couple of weeks. When I say complete sequence, we don't really need the complete sequence. A complete sequence is about half of the genome, the one that is composed of no repeats. The repetitive part of the genome, we cannot map it after the sequence, and a few people care at this point about this. So when we say complete sequence, we talk about a megabase and 1.5 billion base pairs of the genome, about half. Just to amuse you, the cost of the genome sequence, which is shown here, from 2001 to 2012, did better than the Moore's law of the informatics, that every 2 years you cut down the cost by half. So in the last 5 years, the cost of the sequence tremendously went down, and it will continue to do so. And if you have the sequences of humans, like I estimate that probably 20,000 humans have been sequenced so far in the protein coding part of the genomes. The first ones are shown here. You see 3 well-recognized faces: Craig Venter, Jim Watson, and Stephen Quake. And the other ones are 2 Asians and 1 African. The number of variants per genome, with the average, was, as expected, about 4 million. And because the Africans, being more ancient than the Europeans, so they have more variability in the genome. They have more or less to the 4 million changes rather than the 3.8 or 3.9 of the rest of us. If you concentrate only on our proteins, the protein coding genes, and you sequence my genome, I will probably have between 10,000 and 12,000 non-synonymous variants that change an amino acid. And premature stop codons, about 100, each one of us. And the frame shift insertions and deletions within the coding region that mess up the frame the reading frame, we have about 250. Most of them in heterozygosity, of course. And in cancer, as you know much better than me, when the cells divide and they make the somatic cells, they accumulate variants, accumulate mutations all the time. Sometimes these mutations are pathogenic and drivers for cancer. So the pancreatic cancers have probably 40 to 80 non-synonymous changes in their somatic cells, in the cancer cells. The champion is the colorectal cancer, 300 to 400, and the next champion is the melanoma. You can have up to 700 or 600 non-synonymous variants in a melanoma genome. How am I doing with time? I hope you keep... What does this mean? 20 minutes? Wow. So if it's 10 minutes, I go... So there are a few things, three important things that we learn from the monogenic disorders in order to apply them to the polygenic phenotypes. We learn that there's a tremendous genetic heterogeneity in disorders. We know that this difficult thing that we call penetrance, and I go into this because penetrance, in my view, is the most important question in the next 20 years. And the modification of the phenotypes throughout the human genome. Let me talk to you about the penetrance that the ethicists call determination, genetic determinism. Here's the genome of somebody who has achondroplasia, short stature. A disorder is due to one variant in this particular gene on human chromosome 4. If that gene changes, everybody on Earth that has one letter change there is short, and most of these people play in the circuses and you see them from generation to generation. It's amazing that one single letter in one of the two alleles produces a disorder 100%. So the determinism here is 100%. That's a rare example. Most of the examples are like the breast cancer in women in that particular gene BRCA1. If a woman has an abnormality in these two nucleotides in her BRCA1 gene, she has a 70% chance in her lifetime to develop breast cancer. A woman in the population has 9% of probability of developing a cancer in her lifetime. So that the statement is probabilistic, 70%, not 100%. So the penetrance is lower. And the penetrance also most of the time changes because of the identity of the rest of the genome. I try to do this pictorially. Say I have this black background genome and somebody else has this white background genome, and you put a mutation here, a variant, you see it much better in this background than in this background, just for you to understand that a pathogenic variant is always relate with the entire genome for the final outcome. So now the hope is that with the genome sequences, we identify the cause of the complex polygenic multifactorial disorders, the common disorders that we have. You see there are many names. Polygenic, we mean many genes involved. Complex, we really don't know. Multifactorial interaction between the genome and the environment. Like in theology, when you don't know, you give many names to things. And we are still in that debate stage of our development because we don't know what is the cause of the polygenic phenotypes. However, the polygenic phenotypes are common. If I take 1,000 individuals, 600 of us or more, they will develop a multifactorial disorder with strong genetic predisposition in our lifetimes. So it's an important problem. Diabetes, atherosclerosis, congenital heart disease, schizophrenia, manic depressive illness, osteoporosis, blah, blah, Alzheimer's disease, senescence, human behavior. And the whole stage for the polygenic disorders was set up by this particular association in Alzheimer's disease. There's a gene that's called apolipoprotein E, and that gene makes a protein, and the protein has three flavors, like ice cream. Has a flavor blue-blue, red-red, or blue-red. according to the amino acid in position 112 and 158. Well, it turns out that all three flavors are very common among us. All of us have all these three flavors. However, the people that have 2 copies of the red flavor, they have 12 times more probability of developing Alzheimer's disease in a lifetime. So that's a risk factor. And because that was identified a long time ago, people said that, it would be very easy to identify the causes or the predispositions for the common disorders. And now that we know a little bit of the genome, then we can go ahead and find the predispositions to everything. Okay? So association studies have been done. In other words, we take the genome, all the genome variants, and we count what's the frequency of a genome variant in an affected population and what's the frequency of the genome variant in the normal population. And if that difference is statistically significant, we declare that that part of the genome is important for this particular disorder. When I studied genetics at the Johns Hopkins University, one of my professors did not like the association studies, and he used to call them pseudogenetics. So with the pseudogenetics, I take it back, good genetics. All these disorders have been studied in the past 5 years. You don't see each one of them. I just give you the names for your amusement. But every phenotype that you can think of, everything, has been studied. Each line, there were about 10,000 patients and 10,000 controls. And for each line cost probably $4 to $5 million. And the genome was sequenced, and places in the genome have been identified that are associated with all these disorders. The bad news is that even after all these exercise using DNA markers to identify predispositions to human disorders, only 5% of the phenotypic variants could be explained collectively. And the relative risks are very low, 1.2 or 1.3 above the general population for a risk factor. So the majority of the functional risk variants using association studies are unknown. However, money talks. There are many companies in the world that can take your DNA and give you predictions. I give you 3 of them, 23andMe, Navigenics, and DecodeMe. You can send your blood or saliva, and they do the analysis, and they give you your risks. Very good companies, very good scientists. They do their best, but their best is not good enough at the moment. I give you some results from my genome. For example, if the average disease risk for age-related macular degeneration, sort of a blindness in old age, is 6% in the population, and using my markers, I have lower risk, 1.2. My risk for Alzheimer's disease, 7.2 in the general population. I'm at 4.9 risk. I'm lucky. I'm not a homozygous for this red allele. And for diabetes, however, I have more risk than the general population, 33.6, 25.7. So for you scientists, this difference is, it's not important. But the question is how people understand and perceive the risks. And all these companies and all these transitional type of medicine right now is based on the risk perception. Well, I'll give you an example from the literature. Can you predict the height of people using all this genetic knowledge so far? When Sir Francis Galton in 1896, he said that if I measure the size of the father and the size of the mother, I add them up and divide by 2, I can predict the size of the offspring. And he was right 40% of the time, remarkably. If you do that today with all the markers that exist, 54 genomic variants, and you do the prediction covers about 5%. Of course, that's understandable, it covers 5%, because we only look at 54 genomic sites, and Galton looked at the entire outcome of the genome, so we're not there yet. However, people are very hyperactive, personalized medicine, you sequence the entire genome, you find all the variants, and here's my friend Steve Quake. He publishes his genome, so that's it's not a secret. And through the genome sequence that he did, he came down from a risk of 2% a priori for myocardial infarction, based on the family history, to 10% after the different risks. You can take this as much as you want, either the positive way or the negative way. And he also knows that if he takes warfarin, an anticoagulant, he needs a reduced dose of a drug that you have to be very careful with the dose. He needs a reduced dose because he's a fast metabolizer, and he found this by his genome sequence. We're not there yet, but you see what's coming. So in the next 3 minutes, I touch upon 2 or 3 ethical issues. Of course, the genome will give you predictions, diagnostics, mechanisms, and if we understand mechanisms, will give you therapy interventions. The messianism of the genome now is that, we know our health capital, we know the risks for the myriad disorders, we know the cause of disorders, we respond to drugs and the life planning. We know our history, because we can trace the variability down to the common ancestor and see where we are in the things. But the daunting difficulty is that there are too many variants, there are too many phenotypes, and this bloody changing environment that changes the interplay between the genomes and the outcomes of disorders. People that will sequence their genomes in the future, they have to follow up their health capital monthly or yearly. The same way when you have your real capital, the financial capital, you go to the Wall Street Journal or your computer and see the value of the stocks every day, and you see how much you're worth. The same thing with the knowledge of the genome. There's a genome news network that tells you every day if a variant has been identified that predisposes you or positively or negatively to a different disorder. Just to amuse you, the doctor goes to the patient, do you have a copy of her DNA sequence? And then after diagnosis, he goes to the pharmacist. The New Yorker did this. It says, here's my sequence, give me the right drug for me. Now, the sequence of the genome has several ethical, legal, and social and financial problems associated with them. And I dwell on 2, the genetic discrimination and the tyranny of knowledge. All the other ones you can legislate and protect the individuals. But these 2 will definitely remain with us for good. First, the tyranny of knowledge, or the therapeutic gap. The tyranny of knowledge was first described in a nice play by an Athenian writer who lived 450 years before the Common Era, and Sophocles. And in Oedipus the King, that you've probably seen the play, Oedipus, as you know, kills his father without knowing it and marries his mother without knowing it, although he has 50% of his genome shared with her. 4 children. And then at the 3rd scene of the play, he realizes that something is wrong with his family. And he goes to the blind seer, Tiresias, say the geneticist. And he asks, what's wrong with my family and me? And Tiresias said, what misery to be wise when wisdom profits nothing. In other words, why do you need to know if you cannot do anything about this? So this in our lingo we call the Tiresias complex. And most of the committees, the medical committees, they say if you cannot do anything, if you cannot treat or do an intervention or change your life to disturb the outcome of this particular disorder, it's better not to know. Not everybody agrees with this though. And there are many people that want to know even for family planning and financial reasons. And the second topic, the discrimination. You know the discrimination very well, but I'll give you an example anyway. Dr. Robert Ritter from the Reich Department of Health in Berlin, he wrote to his funding agency, the German Association for Scientific Research, in 1941, that from the research that I did and the phenotypic analysis that I did with my helper, Eva Justin, I identified 23,822 gypsy cases that have been conclusively clarified. And then we read at the minutes of the Nuremberg trials that 20,943 gypsies were registered to Auschwitz, please see how close that number is to the diagnostic group of Dr. Ritter. And then 3,461 of those were transferred to other camps because they consented, quote unquote, to voluntary sterilization. And the rest died by starvation, disease, or gas. What's the take-home message from this? That we need to proceed with caution. And now we're more intelligent and we know history, of course, and we're better than the previous generations and we do better. But have that in mind all the time. That's always, the discrimination is always with us. And now the discrimination about the next generation. I give you an example from my clinic, our clinic, a real example. These two people came to the clinic, en français, Monsieur Hazard and Madame Roulette. They're both physicians. Madame Roulette was pregnant in his fourth pregnancy. She was 40 years old. And she came because she wanted to have a prenatal diagnosis for trisomy 21. Fair enough because of her age. She told us that her father had Huntington's disease, a monogenic dominant disorder. So she's at risk for this, and that is a risk. So second disorder. She also told us that her mother, her sister, developed breast cancer at the age of 30, 35. So there's a high probability that there is BRCA1 mutation in this family, so that fetus is at risk. The husband, Monsieur Hazard, being in laboratory medicine, he measured the E4 flavor of his protein. So he's E4, so he's at risk for early onset Alzheimer's disease, and so does his fetus. His nephew has Fragile X disease, a mental retardation due to an abnormality in the X chromosome. So he's at risk for being a carrier, and that fetus is at risk too. And the brother of his father had a bilateral absence of vas deferens, the plumbing of the sperm did not work well, it was not developed well, and then because he has cystic fibrosis, mild cystic fibrosis, so he's a carrier, probably. He may be a carrier, and so is fetus. So the fetus here is at risk for 6 genetic disorders. Now, you may think that this is rare. Of course, it's rare today. But when we know variants in all the 20,000 protein-coding genes, plus all the 15,000 of the long non-coding RNAs and all the other functional elements, each pregnancy will be at risk for many deleterious variants. So we need to rethink the way we practice fetal diagnosis in the future and have clear black lines between what can be tested and what cannot be tested. And with this, I thank you very much. I remind you what the geneticist will be. It will be like a health advisor, analogous to the financial advisor, to protect the health capital, to minimize the effect of unfavorable alleles, and to maximize the effect of favorable alleles. And I'd like to remind you again, for those of you who are in the younger generation, that what it was past is a prologue. And thank you very much again.
Thank you very, very much for this inspiring lecture. I could have listened 1 hour more.